Assalamu alaikum. My name is Nida Nadeem. I am here to represent the topic tissue culturing steps. What is tissue culturing? Tissue culture is a technique in which small tissue pieces or organs are removed from a donor plant cultured aesthetically on a nutrient medium. As described in the picture that there is a parent plant from which the tissue sample is taken and then grown into a medium having the nutrients which are required. Then the sample developed into tiny plants and then plants are grown in a favorable environment. What is the process of tissue culturing technique and what are the steps involved in which? Tissue cultures involves the use of small pieces of plant tissues called explants which are cultured in a nutrient medium under sterile conditions. Using the appropriate growing condition for each explant type, plant can be induced to rapidly produce new shoots and with the addition of suitable hormones, new roots. What is the steps included in tissue culturing technique? There are three main steps included in it, named as initiation stage, multiplication stage, and root formation stage. What is initiation stage? Initiation stage is the first stage of tissue culturing technique. Here, the tissue of interest is obtained and introduced and sterilized in order to prevent any microorganism from negatively affecting the process. It is during the stage that the tissue is initiated into culture. The next stage is the multiplication stage. The multiplication stage is the second step of tissue culture where the in vitro plant material is divided and then introduced into the medium. There, the medium is composed of appropriate components for growth, including regulators and nutrients. These are responsible for proliferation of the tissue and the production of multiple shoots. The last and the third step of tissue culture is root formation stage. Root formation stage is the last stage. Their roots are formed. Here, hormones are required in order to induce rooting and consequently complete plantlets. There is also a general process for plant tissue culture where we have need three basic points. What is these points? Let's discuss. Medium preparation, plant preparations and transferring the plant material to a tissue culture medium. Media preparation. The approximate mixture is mixed with distilled water and stirred while adding the appropriate amount of sugar and sugar mixture. Here, sodium hydroxide or hydrochloric acid is used to adjust the pH. Contents used here will depend on the plant to be cultured and the number of tissue to be cultured. Agar is added to the medium heat and stirred to dissolve. After cooling, the warm medium is poured into polycarbonate tubes. With lids sitting on the tubes are placed in pressure cooker and sterilized for 20 minutes. Plant preparation. Cut the plant part into small pieces. On the other hand, such parts are the African violet leaves can be used as a whole. Using detergent and water, wash the plant part for about 20 minutes. Transfer the plant part into sterilized colored solution. Shake for a minute and leave to soak for 20 minutes. Using a lid, gently discard the colors and retain the plant part in the container and then cap the container. The third is transferring the plant material to a tissue culture medium. For this, 70% alcohol should be used for the sterilization of the equipment used and containers. Open the container and pour sterile water to cover half the container. Cover a sterile lid again and shake the container for 2 to 3 minutes in order to wash the tissue and remove the bleach. Pour the water and repeat this 3 times using sterilized gloves. Removes the plant part from the container and onto a sterile petri dish. Using a sterile blade, cut the plant's material to smaller pieces of about 2 to 3 mm across, avoiding the parts that have been damaged by bleach. Using sterile forceps, place a section of the plant to the medium, replace the lid cap and close tightly. After all of this process, The result in the development of callus, which when produce shoots after a few weeks. Once the shoot develops, then the plant section may be placed in the right environment. There is well lit and warmth for further growth. Here the tissue culture steps completed. Thank you very much.